Hello everyone, my name is William. I'm Maverick.
I'm CHE338.
And today we're going to be presenting about meningococcal disease, specifically the ACWY conjugate.
And B recombinant.
Yeah, and B recombinant vaccine. So the bacteria of focus is called Neisseria meningitidis. The infection causes meningitis, which involves the inflammation of cerebral fluid tissue. It causes sepsis, and it causes disseminated intravascular coagulation. If it is not treated and you are not vaccinated, it could be serious or life-threatening and results typically in around fatality around 70% of the time. It spreads through the exchange of respiratory fluid and throat secretions, like saliva, and it is more prevalent in vulnerable populations with compromised immune systems or in populations who live in close quarters with each other. For example, college campuses where college students live in dorms with each other. So this is just a biology background. So N. meningitidis is a gram-negative bacteria. It is a diploid bacteria, which means it has this sort of two spherical components to each bacteria. And there are multiple strains. The strains are divided into 12 serogroups, which I will cover the component later. But these 6 serogroups, which are A, B, C, W, X, and Y, are the most severe and the most prominent among the meningitidis strains. So this is a picture of the membrane of N. meningitidis. There is a cytoplasmic membrane and an outer membrane, but what's of most important for our sake is the capsular polysaccharide exomembrane. This is the part in which the bacteria is classified by, or the serogroups. These capsular polysaccharide membranes are composed of monosaccharides and here. So as I was saying, here's some of the structures of the serogroup polysaccharide groups. There's A, B, C, W, Y, and X. Basically, they just differ in monomers. Most of them consist of glucose rings with acyl groups attached to it. Mainly, it's just minor changes in the structure. However, these 6 polysaccharide groups prevent against macrophage consummation and acyl groups, but they are just slightly modified, like Men-W and Men-Y's membrane are very similar. Men-A has a phosphate group attached to it. Men-B is the most severe form of meningitis. It has kind of a linear glucose chain. The reason for the difference is not covered in this presentation, as it is outside the scope of the presentation. But anyways, the infection route of meningitis, it enters your mouth or nasal cavity, and from there the virus or the bacteria enters the bloodstream. And then the bloodstream carries it up into kind of the skull or brain area, where it crosses the blood-brain barrier into the cerebral fluid. And in the cerebral fluid, it reproduces and flourishes because the conditions are quite perfect there for the bacteria. And that reproduction leads to inflammation in the cerebral spinal fluid, which causes severe headaches and very severe symptoms that could result in death. So the challenge and goal of the N. meningitidis outbreak is to develop a vaccine that is not only effective, but it is effective against multiple or all of the serogroups. It also has to be effective enough to last multiple years, and yeah, that's what we're going to cover next.
To solve this challenge of meningitis, there are two main vaccines that are used, a conjugate and a recombinant. Conjugate is a quadrivalent vaccine, so it protects against 4 serogroups, A, C, W, and Y, and it offers 5 years of protection, which is why it's more commonly used outside of America. This usually provides improved memory responses for the immune system, so it's better able to deal with it even after the 5 years of protection. However, it does usually require booster doses for those in the age range of administration. The recombinant vaccine is monovalent, so it only protects against serogroup B, and it only provides 1 to 2 years of antibody lifespan. However, this is a lot more efficient than the conjugated vaccine and usually provides a better level of protection while the antibodies are still alive, but it does run the risk of possible mutations, which has only been in a few cases. To dive more into these vaccines, the first one is the meningococcal conjugate vaccines. These are usually administered between 11 and 12 years old for the first one, and the booster at 16. The brands of these include Menactra, Menveo, and Menquadvi. Since these have been recommended in 2005 by the CDC, we've seen a 90% decrease in meningitis cases. And then the other main vaccine used to solve meningitis is the serogroup B meningococcal vaccines, which is administered between 16 to 23 years old and requires multiple doses, which is just coming into the doctor's office a couple of times. The brands that this includes is Bexsero and Truminba. These kind of popped up in 2014 when they first got approval for widespread use. To kind of dive into this newer one, Truminba, it is a recombinant vaccine, and is specifically a subunit vaccine, which means it includes 2 lipidated factor H binding protein variants, which we'll talk about how it's produced later. These work by attacking the protein components of the bacteria, not the sugar coat, so that the body is better able to recognize it due to the modified protein components. Then the recombinant part of this vaccine means that it's not produced from meningococci bacteria, and is instead produced from a different bacteria, which is E. coli, as we'll dive into this in the next slide. This figure kind of shows how the vaccine works in the body. It's put into the body, then it enhances the target protein, the adjuvant enhancer hits the target protein, where then the antigen-presenting cells, they will be recognized, and then the immune system can better detect the threat due to the enhancement. To produce Truminba, it is made by making the 2 FHBV variants individually, and it's made from an E. coli bacteria. This is made by growing in a fermentation growth media until it's at a specific density, and they take it out and they purify it through column chromatography, so it's just the FHBP variants that they want. Then they add polysorbate 80 as a preservative to make sure that it can sit on the shelf until it's needed. And this vaccine then is composed of 60 micrograms of each of the FHBP variants that they want. Then they add the 0.018 milligrams of PS80 for, again, preservation. And then they add 0.25 milligrams of aluminum phosphate and 10 micromolar saline. That's kind of just the medium that the vaccine goes into, so it's able to go into your bloodstream. This figure right here shows this process, though for another vaccine, and kind of how this process goes and the two playing together.
So multiple vaccines have been kind of released lately that addresses some of the concerns covered earlier, both conjugate and recombinant. Sanofi got approval for a conjugate vaccine. That is the only vaccine in the US that uses a tetanus-based toxin as the protein carrier. And it covers a very wide range of ages from 2 all the way to 56. And Pfizer also got approval for one of their recombinant vaccines that is used for persons age 1 through 7. And while there are a variety of vaccine manufacturers and products, right now we'll only be covering the conjugate vaccine that is manufactured by Sanofi, which is first administered to adolescents at age 11, then a booster at age 16 is administered. However, if the patient is 2 years old or under and have the following immune compromising conditions, a 2-dose primary series is administered 2 months apart. And with regards to this market data over the past couple of years, in 2011, the meningococcal vaccine kind of saw a $2.88 billion market cap, and this is expected to reach $5.07 billion by 2030, and this is due to a number of reasons, such as expanded research and development, kind of awareness and programs that push for more vaccine rollout, as well as overall population increase. And the conjugate vaccine is a dominant segment just because it has the ability to kind of last longer, the immunity lasts longer, as well as the ability to induce herd immunity. And the primary end users is pediatrics, followed by adults. And this is some data from the CDC that kind of tracks the vaccine progress, or like the number of cases over time. And the first vaccine was introduced in 1978, which you can see is here. However, it took a while for the cases to kind of see a noticeable decrease just because of vaccine rollout, campaigns and policy. The public kind of just wasn't aware that this was a thing. And here we're going to cover kind of like a research paper that discusses, in case an outbreak occurs on a college campus, the cost overview. And for a rough introduction, this was based off the outbreak in 2015 in Providence College, and they tracked costs over an 18-month period. And the serogroup B is the primary serogroup that was kind of targeted in this study. But kind of it's not just that. This serogroup is also responsible for 60% of invasive meningococcal cases in the U.S. among young adults, and accounts for all 10 college outbreak cases since 2011 to 2017, in which there were 42 cases and 2 deaths. And the unique part about this study is that they don't just focus on vaccine costs, they also focus on, say, staff, other resources, quarantine, things like that. And what the data and kind of pointed to was there should be a focus more on vaccination in a non-outbreak setting rather than actually responding to the outbreak as expected. However, one point is that the cost of the outbreak does vary widely, from $50,000 to almost $650,000, just because of campus population size, the size of the targeted population, whether or not the government or local or federal provides aid, whether or not there is in-house support and volunteers. All those will really affect that cost.